Hi everyone, welcome to the FDA webinar on Identification of Medicinal Products, Path to Global Implementation. My name is Ron Fitzmartin. I work in the Center for Biologics Evaluation and Research. Our speakers are TJ Chen, Taylor Priya, and Larry Callahan. Today we are going to give you an update on FDA's approach to the implementation of ISO standards for IDMP. As you may know, the ISO IDMP standards were developed many years ago. And now, as the international community is focused on implementation, we have seen the need to address some issues with the standards so that they can be fit for global implementation. Our first speaker is TJ Chen. He will provide an update on the work we have been doing with ISO TC215 Workgroup 6 and WHO UMC to address the issues with the use of management as a control vocabulary management. Pharmaceutical. Now, I will turn it over to TJ.
Thank you, Ron. Today, we're going to discuss the ISO Identification of Medicinal Products, IDMP standards very briefly, and focus on the effort to harmonize the regional dose form for the generation of global pharmaceutical product identification, PhPID. The Identification of Medicinal Product, IDMP, is a suite of 5 related ISO standards that are used together to define the data elements and structure to uniquely and unambiguously identify medicinal product, pharmaceutical product, and substance. The standard promotes common vocabulary to improve people communication. It also promotes common message standard to improve IT system communication. The 5 standards each focus in a particular area. 11615 define medicinal product and specify the identification at medicinal product level as NPID and packaged medicinal product as PCID. 11616 include algorithm to calculate pharmaceutical product identifications, PhPIDs, based on a combination of substance, strength with unit, and dose form. 11238 defines substance. Larry and Tyler will get into the detail about this standard in the second half of the webinar. 11239 defines terminology for pharmaceutical dose form, units of presentation, and routes of administration. And 11240 specifies units of measurement for medicinal product. With well-defined data elements, common vocabulary, and standardized electronic message, it improves the data quality. It also makes it easier to share information across jurisdictions when the standard is implemented in multiple regions or globally. And with that, it improves our ability to identify, assess, and respond to patient safety or medication incidents. And the standard also makes it easier to find alternative products to mitigate product shortage. It also makes it easier to compare similar products in a region or across jurisdiction. However, to maximize these benefits, cross-regions or global agreement on common substance ID and dose form is needed. First, let's look at how ISO IDMP enables us to connect medicinal products together in a region and across region. Here we have FDS liquid gel, which is a medicinal product that is authorized to be marketed in the United States as OTC product. The barcode on the right is the NDC code, which is the US implementation of medicinal product identification at package level, PCID. Each capsule is a pharmaceutical product that contains ibuprofen at 200 mg. With the information of substance, strength, and dose form, we can calculate a level 4 PhPID, which would allow us to connect to Midol or the generic ibuprofen in the United States. With cross-region agreement on the dose form and substance ID, we can also connect to Nurofen in Europe and Ringo in Japan. Because all these products share similar pharmaceutical composition, So when one of the products causes an adverse event in a region, we know other product will cause the same or similar adverse event in that region and in other regions. And with that, we can build a global pharmacovigilance. And also we know that when there's a product shortage with one of these products, the standard enable us to easily identify alternative, and that's how we can mitigate product shortage across regions. However, I'd like to bring your attention to the dose form. Each region can assign dose form at different granularity. We have capsule for Advil in the United States, and the Nurofen has soft capsule in Europe. Based on the current ISO IDMP standard, we will not be able to generate identical level 4 PhPID to connect these two products together, which poses a challenge to the pharmacovigilance at global level. So there are efforts trying to harmonize the representation of those forms so that we can generate identical level 4 PhPID to connect these products together. ISO 11616 defines PhPID as a set of 4 labels based on the combination of substance, strength, and dose form, and the label 4 being the most precise label. As we can see here, substance is a key for all PhPIDs. Without globally agreed substance ID, there is no global PhPID. And also, without global consensus on dose form ID, there is no level 3 and level 4 PhPID. Before I go to the next slide, I'd like to get your attention on the term administrable dose form for level 3 and level 4, because the term carries some meaning that we will discuss in the upcoming slides. Current ISO PhPID standard is based on the assumption that there are central codes for substance ID and dose form. However, there's no agreed central code for either one as of today. There are efforts trying to get to global substance ID, and then we will get into the detail later. I'm going to focus on the effort to harmonize regional dose form for the generation of global PhPID. In 2018, we mapped the 166 FDA dose form term to the 490 EDQM term, which is used in Europe, hoping that we can get some decent percentage of mapping. But it turned out that we can only map 37 terms 1-to-1, and that does not even factor in the definition. If we include definition, the number is going to be even lower. And just to make sure that this is not a unique case, we also map the SNOMED CT, CDER terminology, and a copy of those forms used by Health Canada in 2018 to the EDQM term. And as you can see, the number is not very encouraging. So we come to a conclusion that region-to-region terminology mapping is not a viable solution. Ron presented this finding and challenge in the spring 2019 ISO meeting to keep the ISO 11239 and the technical spec 20440 open for review and revision. In 2020, the UNIQUENNESS project, which is the EU effort to implement IDMP, presented a gap analysis report during the ISO meeting, and the report stated that the adoption of this IDMP standard has been difficult at times. National competence authorities are facing backward compatibility issues because the granularity of the terminology varies frequently from EDQM. It also pointed out that US FDA has shared implementation difficulties. So the challenge is not localized to a single implementation. Some improvement and revision to the standards are necessary to make it easier to adopt and implement. The ISO 11239 specifies that each pharmaceutical dose form term will have 6 associated characteristics, and those are state of matter, basic dose form, transformation, release characteristic, intended site, and administration method. The EDQM implemented ISO 11239 and established robust terminology for each of the characteristics. the characteristic is not as granular as a pharmaceutical dose form term. So the first proposal is to use the collection of the characteristic instead of the pharmaceutical dose form term as an input to generate a global PhPID. So with the example we had earlier on capsule and subcapsule, here on the left-hand side we have 4 pharmaceutical dose forms: soft capsule, hard capsule, plain old capsule, and capsule that are gelatin coated. And as we can see that all these 4 pharmaceutical dose forms share exact same 6 characteristics. They are all solid with basic dose form as capsule, no transformation needed, conventional release, infanticide is oral, and administered by swallowing. So instead of use the pharmaceutical dose form terms to generate 2 incompatible level 4 PhPIDs, we now suggest to use the collection of the codes associated with a characteristic as input to generate global PhPID, which would then allow us to connect products in soft capsule, capsule, hard capsule, and gelatin-coated capsule together. And with that, we now can connect the alveolar capsule to the soft capsule neurophil. This approach would allow each region continue to use the regional dose form term and at the same time to generate global PhPID without a central dose form system. The approach was presented during the November 2019 ISO meeting and was well received. In May 2020, the ISO working group established a subgroup to revise the 11239 and the related TS 20440 to include the characteristic approach in the standards. And during the revision, we encountered another challenge with products that require transformation. The example here is powder for solution for injection that need to be dissolved into solution for injection to administer to a subject. The powder for solution for injection is classified as manufactured dose form based on the ISO standard. And the solution for injection is the administrable dose form. And as we discussed earlier, the ISO 11616 utilizes the administrable dose form to generate level 3 and level 4 PhPIDs. And with that, we introduce the basic administrable dose form characteristic to capture the basic dose form that is administered to a subject. So let's look at the table here. The first row, powder for solution for injection, started as a powder that need to be dissolved into solution for injection. And the second pharmaceutical dose form, concentrate for solution for injection, started as a liquid concentrate that need to be diluted into solution for injection. And the third one, solution for injection, does not need any transformation. So the basic administrable dose form equal to the basic dose form. So let's look at the 4 columns on the right. We know that the 4 characteristics together is sufficient and adequate to represent the administrable dose form. The original 6 characteristics are actually associated with the manufactured dose form. So with that, we propose to use the 4 characteristics, the 4 columns on the right, as input to generate global PhPID, which would then allow us to connect medicinal product with same administrable dose form together, regardless of its original manufactured dose form. This approach was presented during the February 2021 ISO meeting. ISO TC215 Workgroup 6 then suggests WHO, UMC, and FDA to conduct a global PhPID pilot with broader medicinal product to evaluate the feasibility of using 4 characteristics to generate global PhPID. FDA then assigned the whole 7 dose form characteristics for about 1,400 US marketed medicinal product based on 34 substances that was identified during the unicorn pilot. UMC then take this data set further validate and refine some of the assignment to generate level 4 PhPID. Together, we perform a data equivalency assessment and report a finding during the ISO 2015 Workgroup 6 meeting in June this year. That was last week. Besides the medicinal product based on the 34 substances as described in the previous slide, the pilot project team also include COVID-19 vaccine in assessment and identified some challenges. The pharmaceutical dose form for the Pfizer COVID-19 varies from region to region. It is noted as dispersion for injection by EMA, suspension by FDA, solution for injection by UK. We were not able to generate identical level for PhPID for the same product. And also the strengths can be expressed in different regions with different units. For the AstraZeneca COVID-19 vaccine, EMA measured by infectious unit, both UK and Australia measured by viral particle. And again, we were not able to generate identical level for PhPID for the same product. The pilot confirms using dose form characteristics as input to generate global PhPID as a viable solution to connect medicinal products in different regions. We also recognize a single organization with global perspective to consistently assign dose form characteristic, strengths, and substance ID is important for global IDMP implementation. The working group will continue the revision of ISO 11239 and technical spec 20440 and also evaluate the impact to other IDMP standards. Before I wrap up the presentation, I'd like to share with you the FDS approach to the IDMP standards. FDA has been using standards similar to IDMP for decades. It is our intention to conform to the international standards so that we can exchange medicinal product information with partners and regulators around the world. And with that, so that we can do pharmacovigilance and to mitigate product shortage at global level. Since ISO published the IDMP standard in 2012, we started to assess the standard we have been using against the ISO standard, and we come to a conclusion that the UCOM code for unit of measurement, unit for the substance, and NTC code for medicinal product identification all conforms to the ISO IDMP. And we are working with experts internationally to try to harmonize those forms for global PhPID. In 2019, we also established an FDA EMA IDMP collaboration framework to promote harmonized IDMP implementation. And we continue to work under ISO TC 215 Workgroup 6 to revise the ISO IDMP standard to make it easier to understand, easier to adapt, and easier to implement. We also started to develop an internal IDMP implementation plan and hoping that by 2022 we can start testing and validate the global PhPID. With that, I'm turning the presentation to Larry and Tyler. Thank you.
Hello, my name's Larry Kellen. I'm with the Office of Health Informatics at FDA, and I'm here to talk today about the ISO 11238 standard, which is the standard for substances, which is part of the IDMP effort, and also the Global Substance Registration System that actually implements the ISO IDMP1 standard. For substances. And again, this has been a collaborative effort primarily between the FDA and the NIH's National Center for Advancing Translational Science, NCAT, and CBG, the Netherlands group, and the EMA. And many others have been involved also. And again, it's all about organizing information. FDA has the most important or valuable repository, human biological product data, but there's limited integration. And some of it's due to the submission process that we get stuff in paper and PDFs, which is always electronic paper. We get them as applications. And again, it's usually not a single submission, but there's supplements and a lot of other things that come in. So it's always hard to keep track and put things together. And the IDMP is an effort to organize this information on a global scale. And the amount of information, of course, is drastically increasing. I mean, there's a lot more drugs and vaccines on the global marketplace. There's rapid screening methods that give us a lot more data than we previously used to get. There's enzyme and receptor profiling, CYP and transporter and receptors as well, and genomics and epigenomic data that we're getting. And then also electronic health record data we get occasionally. And also there's many CMC changes over the life cycle of a drug, and to track them all back and to get them into databases so we know what's actually in the products is challenging. So again, it's the marketplace. Of course, it's a global marketplace, and pharmaceutical products, of course, are the highest value products that we have in the marketplace. They're highly regulated. There's small amounts of material, relatively low shipping costs, and then there's multiple ingredients from a variety of companies that make up the product. There's very few companies that are completely vertically integrated. So you have active ingredients coming from one company, excipients coming from other companies, starting materials from other companies, packaging, testing, and reagents. A lot of this stuff is all done by separate companies, and shortages in any of these areas can result in the product not getting on the market. And again, it's a global pharmaceutical marketplace, as we said. You know, if you look at APIs, this is a slide from a couple of years ago that CDER put together, and it has, you know, it basically shows that the U.S. only 28% of the APIs that are in our business, so products come from the U.S., and the other ones come from China, Germany, Europe, India, and the rest of the world. So again, it's only a small percentage that's come from the U.S., so it's really a global marketplace that we're dealing with. And again, this shows the pharmaceutical supply chain. And again, you start with raw chemicals oftentimes that get the key starting materials. And again, this is really where it starts off at FDA, where we'd like to know what are the starting materials that make up a drug, that make up the active pharmaceutical ingredient, and we want to capture that. We want to capture the formulation. We also want to capture the product packaging and labeling. And again, substances come in on many of these areas. And then once it's marketed, we want to track the drug and see what it's actually doing, and who's distributing it, how much is getting distributed, and what's the health outcomes from the use of the medication. And again, substances are one of the key linchpins for organizing this information. And again, names, of course, are insufficient to describe substances. The same name can lead to very different substances. One example is lime, of course, which is a fruit, and then lime, which is a chemical that you use for cleaning and other things. And then different names can be the same substance. Again, acetaminophen is quite a bit different than paracetamol, but they're basically the same substance. One name used in the U.S., one name used in Europe. And again, we want to define substances not based on names or terminology, but based on core scientific principles, and assign a permanent unique ingredient identifier to each substance. And that's called the Unicode at the FDA. And it's basically it's a 9 random character and then 1 check character. So it's always a 10-character long entity that describes, that's permanently tied to the substance. And again, when we look at substances and we say, well, you know, how do you describe substances? Well, it's something that Aristotle dealt with. And again, we've taken a very similar approach because, again, we have more than just chemicals. I mean, if it was just chemicals we're describing, it would be pretty straightforward. We get the molecular formula, but we have a lot of times we have, you know, modified cells, modified organisms, modified organs. And so to define those substances, we really have to go to a broader definition. So it's anything that has matter and capable of separate existence are the core principles that we went with when we were defining substances. And again, it's something that's basically a unit of matter that can be quantitatively measured. That's what we consider a substance to be. And we looked at substances and we divided them into 5 groups. Everyone goes in at least one box. You can certainly be in more than one box, but you have to go in one box. And you can be a chemical, you can be a protein or peptide, you can be a nucleic acid, you can be a polymer, or you can be structurally diverse material. And we assign each substance into one of these boxes. We can have alternative definitions. For instance, a short peptide could be both a protein and a chemical. And so again, the system is flexible enough to handle that. And again, we don't describe substances based on their use. We don't care how they're used. It's really what we think they are that's important. And the same substance can be manufactured or isolated using different methods. For example, salmon calcitonin can be made synthetically, or it could be made recombinantly, and it would be the same substance to us because you end up with the same sequence of a protein. And again, these 5 elements that we have basically things that are monodispersed, meaning they're pretty much a core of one thing. We're not taking into account the impurities and other things, but chemicals are pretty much a single entity, and we define them based on their molecular structure, which is their connectivity and their stereochemistry. Proteins we define by their amino acid sequence and type of glycosylation, modification, and any modifications that come onto it. Like if you attached a chelating agent to the protein, we would capture that chelating agent along with the metal that you have for that. And again, nucleic acids, we would capture the sequence and the linkage and any modifications as well. And again, these are all monodispersed chemicals. Polydispersed would be polymers, and polymers are sometimes some of the most difficult things for us to describe. We have to have the type. Is it a monopolymer or copolymer? What geometry does it have? Is it linear or branched? Is it a type of copolymer? Is it block or random? We capture the ratio of monomers, the modifications, the molecular weight or properties such as viscosity related to molecular weight. And we also capture the biological source for many of the biopolymers. So polymers are some of the most difficult things for us to describe, and but our system does handle it. And then we have structurally diverse material, which are viruses, cells, tissues, complex material, and that's predominantly by taxonomic, anatomical, fractionation patterns, sometimes physical properties, and again, any modifications that end up with permanent changes in the actual structure of the entity. And again, the scope of substances is very broad for medicinal products. It can be active ingredient, inactive, or excipients, impurities, metabolites, targets, off targets, and even processing materials and starting materials and things like that. So again, once we define substances in the scope, there's also a need for higher level information. A lot of ingredients are multiple substance materials. Simethicone is a perfect example where you have basically sand and dimethicone mixed together to form a substance or form an ingredient. And colorants usually are more than one actual molecular entity, and flavors as well. So we need to have a level, and we call it specified substance group 1, that takes this into account. It also describes extracts, and it distinguishes things like polymorphs. If you had two different polymorphs, they're the same substance, but they'd be different at the level of specified substance group 1. And then for things like cellulose, by the flowability and compressibility, they may differ, and they'd be different. They'd be the same substance again because they're cellulose, or microcrystalline cellulose, but they would differ by specified substance group 1 depending on their flowability or compressibility. And again, this is something we're just starting to implement at FDA. And then there's a need to also tie it to manufacture in a process, and that's what we've done, what we call specified substance group 2, where we tie material to the actual manufacturer, or group 4, where we tie it to its actually manufacturing process. And so that's, those are the other levels that the standard has developed. And we need to tie material to a specific grade, which will be USP grade or EP grade. And again, that can be something SSG2 can be SSG3 too, so we would capture them both together. And again, there's a need for specification information too, and that would be at SS group 4. And a need to obtain information on processing materials. Again, that's at SSG4. And there's a need to establish and monitor the supply chain, which SSG2 would help us with. Again, what we've done in version 2 of the ISO standard is we divided manufacturing specifications at the group 4 level, so they're separate entities and can be captured separately. Again, this is just a screenshot of the actual model that we have. And again, everything sort of goes back to a substance. You have to have one of these things, and you get a version. And a substance can lead to multiple specified substances, or it could lead to multiple group 1 or group 2 specified substance. And then again, group 4, we capture all the modifications and how they're made, and also the specifications at group 4. Again, the implementation, group 1, we've already started to implement it. We're still working on capturing the details of glycosylation. So for like proteins, the actual details of glycosylation, how much of a given glycol form you have on a protein, that's going to be captured at the group 1 level. And we're still working out trying to get a glycan library so we can capture that data. At group 2, we need a common identifier for companies to implement it. Right now, there is no common identifier for companies. In the U.S., we use a DUNS number, and in Europe, they use the EU, the EMA has their organizational database, which gives it a separate number. We also have an FEI number at FDA, so we have 2 different identifiers, and what we want is a single identifier to tie it to a company and then to keep track of it. And specification module is being developed too, and we already have developed an impurity module with USB is under development, so we can capture at least the pharmacopoeial specifications in sort of an organized manner. And we've also developed a manufacturing prototype that we're looking at, working with our PQCMC colleagues at FDA to see if we can implement it. And again, so we think the global marketplace for ingredients requires a global system to monitor the global supply chain. That's what the decision made at the IDMP level, that there would be a global substance ID. The global repository would contain regulatory information and data on ingredients. It would help address shortages, substandard and counterfeit materials, and perhaps even coordinate inspections. FDA with NIH, we've developed a software system that can be used by regulatory agencies and companies and really anybody throughout the world to register substances. And the good thing about this system, it not only has chemicals, it handles proteins, nucleic acids, and then organisms and polymers. And it's one of the few systems that handles it all together in one entity. So that's what we're very excited about, and hopefully more people will start using it. And the system is being used by the EMA and other European agencies like BfArM in Germany, and also the WHO UMC group, who we're working with to try to develop a common substance identifier. And we think having a common system will make it easier to transfer data and allow for more readily international implementation. And again, we still haven't agreed on a global substance ID. At one point, it was going to be the Unicode, which is what the FDA uses, which again is a 9 random character with 1 check character identifier, alphanumeric identifier. But again, that hasn't been agreed upon. In the global substance ID, there's still talk whether we need a single organization to contain a global substance ID, which we probably need to do this work. And then we're working with UMC. Is underway with WHO UMC is underway to sort of work on what could be a global substance ID that they may maintain. Since they deal with countries throughout the world, they're probably in a better place to maintain an actual global ID than we are at FDA. And of course, the global ID is needed to implement the global PhPID, which TJ talked about in the previous slide. And again, do we want a single organization to handle all global IDs, or do we want a federated approach? That's still something that needs to be decided. So what is the GSRS? Well, it's a software system, and what it does is it registers and defines all substances that the ISO standard has delineated, and it basically assigns a unique identifier with it for each thing, and it's compliant with the ISO IDMP standard. It's the only system out there that currently is compliant with the standard, and again, it's something that we developed with NCATs, and we developed it kind of in sync with the standard, that we modified the standard to make sure that the standard was implementable. And what it does at FDA is it ties substances to products, applications, clinical trials, adverse events, and drug targets. And we also have limited quality information in it. And again, we have some LADMER data, we have some metabolites, we have relationships between metabolites, CYP enzymes, and transporters, and some limited pharmacokinetic information in it as well. Again, it works in all modern browser. It's something that's freely distributed, that's cost-free. It's through NCATs, and they distribute it with a large set of public domain data, and it's updated periodically, usually on a monthly or bi-monthly basis. It links to many outside sources. It has structure sequencing-based searching, faceted, advanced field-based searching, and the data is downloadable in a variety of formats. There's approximately, current status at FDA, there's approximately 200,000 substances registered. About 120 have been curated, and the way they get curated is that one person enters a substance, another person, usually a senior scientist, will look at the substance and agree that it looks correct, and then only then is it assigned an ID, a permanent ID. And we have over 2 million names and codes that are associated with substances, and nearly 200,000 relationships to drug targets, and much of the data is in the public domain. And how it's used at FDA, again, it integrates a lot of different internal databases at the agency. The adverse events, we get product information through SPL, since SPL requires a Unicode for all the ingredients listed in a product. So we basically link to all the product information for really nothing because of SPL. We also link to applications and NDAs and INDs where we actually enter that data by hand oftentimes, which takes a lot of time, and to tie it to an IND and an NDA or BLA. And then we also by hand tie it to clinical trials oftentimes if we see a clinical trials. In the future, we hope that the GSRS will be used to facilitate digital submissions of formulation, quality, and pharmacology data, so we don't have to do all this stuff right now for applications and other things. And we also have a number of classification systems in the FDA, including CFR, DEA, ATC, and other classification systems that are used throughout the world. And again, we have another initiative that we're working now with the Pistoia Alliance, and that's to develop data standards for in vitro pharmacology data. Like I mentioned before, we have some of this in the GSRS now, but we're entering all of it by hand. So we want to get a data format where it will come in automatically into systems like the GSRS, and we don't have to do the data enter. We're able to capture the data, and hopefully reviewers will be able to use it more facilely. And we're setting up teams, and we hope to have quick development with it in sync with the GSRS. We also have a vaccine initiative, and again, I don't need to tell you that vaccines are the most important public health tool we have. They can literally save the world as they're doing now, but there's no international nomenclature. Names vary significantly throughout the world. As TJ showed, the product names, you'd think there were 3 different products for the Pfizer vaccine because of the way they're named throughout the world, but they're really the same product. So, you really need international nomenclature for this thing to sort things out. WHUMC has set up a site for registration of vaccine ingredients and related substances, and we're using it now to work out common controlled vocabulary, possibly a global identifier, and we hope to have a pilot somewhat complete by September to know if we should go forward and then get industry involvement also at some point. So these are the public resources, and again, Tyler will go over them in more detail than I have here, but if you want to find the software and data, you can find it at the NCAT site. If you want a list of all the Unicodes, you can go to NLM, although that's going to probably come down at the end of the year since our collaboration with them is ending. We have a Genus meetings. We've had them at USP. We've had them at WHUMC. We've had them at CBG. And we have those usually annual meetings. We haven't had one since the pandemic broke out, but we should have one soon once we release version 3.0, which Tyler may touch on too. And you can get on the Genus notification list by going to the NCAT site and registering. Again, this has been a real collaborative effort with a number of parties involved, and this just illustrates some of them. And again, it's a global effort, and we really enjoyed working with all the people, and we've had valuable contributions from each of these organizations. And again, this just shows the FDA team and the current NCATS team. These are the IDMP members, and there's some members that I missed, and I apologize to that. But again, there's been a lot of foreign regulatory participation in this. And we've worked with Kew Gardens on how to represent plants, and we've worked with USP with impurities and other things, and of course with UMC with group and things like that. We have a good team at NCATS and at FDA, and it's been a real pleasure working on this project. And we think it's an important project, and again, we think the whole industry will benefit from it because again, it's something that's freely distributable. That these systems can cost millions of dollars to buy, and we're giving it away for nothing. And so, particularly for small companies, it makes a lot of sense to use a system like ours since it's basically cost free. And again, that's the end of my presentation. I'd like to thank you for listening.
Thank you, Larry. Hello, my name is Tyler Perrier. I'm a chem informatician with FDA and the Office of Health Informatics, and I'm discussing the GSRS open software today. Before I start, it's really important to note that the GSRS open software is a collaborative effort with a lot of organizations, but principally with the National Center for Advancing Translational Sciences, NCATS at NIH. They really have been the spearhead. They've been spearheading the effort since the FDA began collaborating with them in 2013 on this effort. So the outline of what I'm going to discuss is I'm going to go through a few different things. I'm going to describe what the software is, how it works, where it's used, where it's going, and then I'm going to give a little bit of detail on how you can get involved. So the first thing is what it is. GSRS software is freely distributable and open source software. It's an implementation of the ISO 11238 standard as part of IDMP, and it's created and maintained by NIH NCATS in collaboration with FDA and several other organizations. On the right hand, you just see some screenshots of the web interface for the GSRS software and database. So the next question is, what is it for? And it's a self-contained web application and database for registering, storing, searching, and exchanging substance information in a machine-readable form in compliance with ISO IDMP 11238 standard. It's freely distributable and can be used as a local substance registration system by regulators, researchers, and industry. And in fact, it can even just be used, locally by yourself if you have need for a personal substance registration system. We know that there are folks out there who just download the software and use it locally to help keep track of substances. So it's part of what you might want, why you might want this to begin with. Just a little bit of background. Larry gave some of this background as well, but there's a wide variety of substances that exist in the marketplace right now, and these have things from complex molecules like monoclonal antibodies with toxin conjugates, as well as like plant-derived substances, animal-derived substances, small molecules, and of course vaccines, which can get quite complex. And it's important, we believe, to have a systematic definition and systematic curation in order to put that data to systematic use. Because once you've agreed on what the substances are, and you can actually give them identifiers and track them, then you can start doing a lot of things systematically. You can do explorations of substance space. You can start keeping better track of metabolism, of inventory, nomenclature. The list goes on. Of course, one of the key areas that we're interested in is in pharmacovigilance. But really, there's a lot that can be done once you agree on what the substances are and how to handle them at a basic level. So, as Larry mentioned, the first thing you have to start with is what really is a substance. This is a slightly modified but still compatible definition with what Larry presented and what's in the ISO standard. Is a substance is a conceptual physical entity that is capable of separate existence and is uniquely definable based on its immutable chemical, physical, and/or taxonomic properties. So it's a real material, but it's the concept of a real material, defined at some level of granularity. There are 5 main substance types at the base level. There are chemicals, polymers, proteins, nucleic acids, and structurally diverse substances, and each of them are defined by different things. And this just gives kind of a basic overview of what those things are, at least a high level. What's important to know is that in the past, really there weren't that many kinds of substances used that often. Most of at least for the APIs coming into FDA, most of the drug substances, those were small molecules. Those are what we call chemicals. You can see in this chart here that's slowly not becoming the case. So in 1984, in the INN list, the International Non-Proprietary Name Lists, almost all new generic names granted were for small molecule substances. But over time, that's really diminished, and it's to the point now that every new list of generic names that are approved by INN, only about half, and in fact less than half, tend to be small molecules. And a good portion of them end up being protein substances, and increasingly structurally diverse and nucleic acid substances as well. So it's important that we get a handle on all of these kinds of substances. We can't just stop the line at chemical small molecules. So to explain what GSRS software is, it's the main task of the GSRS software is to help in registering a substance record and associating relevant data to it. So you might have data for your substance in PDFs, in other databases, in Excel sheets, on web pages, or maybe even as images of the chemical structures. And what GSRS is attempting to do is to give a place to systematically put that data into a structured, machine-readable, searchable, and exchangeable model that you can use for making for discussing the substance information, but also just for creating unique identifiers and tagging important information to that substance. So in order to accomplish this, GSRS has several tools that are helpful in that registration process. There are data entry forms that are part of the web interface. There's a name to structure tool. There's an image to structure tool. There are other tools that are specifically for other substance classes as well, things that will help translate protein sequences and so on. There are configurable validation rules to have certain to ensure that things that look like they could be duplicates or be incompletely defined can be handled. Same thing. There's a uniqueness check algorithm that's helpful in comparing different substances together to make sure that they are indeed unique. And there's audit and edit history as well inside of the software built in. It's now time for a small demo, which I will attempt to do on the next slide. I want to show a little bit of what it looks like to register a substance in GSRS. So in this case, I've downloaded the GSRS instance. From the NCAT's website, you can download the source code, and you could install it on a local machine, or usually on some sort of server, and then it's self-contained, just by itself. It sets up a database, and then from that database, it gives you a little web interface. So when you log into the web interface and you log in as a user, then you can decide what you want to do, if you want to search or browse the stuff you've already registered, or maybe you just want to register something new. So in this case, I'm going to show off registering what it looks like to register a new substance. So as mentioned before, there are different kinds of substances, but we'll start with the most common one now, which is a chemical. So this is the registration form. There's a lot of things that you can do inside it. You can add to a given substance record. One of them, you can start by just saying some basic information about the level of the definition. Usually it's going to be primary definition type, and it's going to be a complete level, but you could have an incomplete level. You can specify what your intended access level is, whether you intend this to be a protected thing or you intend this to be something that's for wider public, and you can add certain tags that you might find interesting. Something like, these are results from, screening library A, or whatever you might want to use. As you're going through things, you also add references for where you got the definition from. That's a very important piece, is for us to know where definitions come from, and that's important for a lot of people who use the software. You can add individual names. There's no limit on the number of names that you might add to a substance, so this form helps you fill those things out. So let's say, for instance, that I am trying to register something called, compound A. Maybe I have this thing known as compound A, but I also have another synonym for this that's called, 1, 2, 3, 4, special compound. You can add many names, and then with them you can decide what their types are. If they're an official name, you can add more information about this official naming organizations that use these names. All of this is in compliance with just the ISO standard. Now, if you happen to have a name, we encourage people when they're using the system to enter all names that are relevant. But if you happen to know a name that is resolvable, that carries meaning, you can actually use a tool to help fill in some of the other information. So, for instance, let's say that I happen to know, I was looking for something like, propane. So if I search for propane, I can click the resolve button, and then I can say, well, here's what these different online databases have said procaine means. I can compare them. In this case, they're all the same. And I can decide if I want to import the structure from one of these to use in my definition. So let's say I'm going to use this one from NCI. Now I have this chemical structure in the chemical editing tool, and if there's some minor modification I need to do to it, I can do it there. I could do something there. I can change the aesthetics, or if I just happen to have a smiles or a mole file, I could import that directly as well. Similarly, so those are some of the tools that you can use just in the system. You can have a registration form. You can enter the names, then you can enter the definition, and then once you enter with the definition, there are a lot of ways to get the definition in. For a chemical substance, you can either import an existing file that you might have for that chemical substance, or you can use the resolve name feature. Sometimes it turns out that you don't have something that is nearly as simple to use, and it might be that what you really have is like a PDF, and inside the PDF somewhere is a chemical substance, chemical structure, and you want to have this registered into your system. This is something we find happens all the time at FDA. So what we need to do is we need to be able to redraw this chemical structure, and typically that's just too much work for us to do. We don't like to do that by hand if we can avoid it. So in the interim, what we've done is we've allowed users to copy a chemical structure as an image. So in this case, I have this as an image. I can just copy it to my clipboard, go back to GSRS, and I can attempt to paste it. And what it will do is it will say, look at this image right here, and it will use some computer vision in order to determine whether this is what the chemical structure would look like as a real editable, machine readable form. So in this case, this now turns into a real chemical structure. It's not perfect, so you always have to compare it back and forth. In this case, it looks like it did pretty well. So we can hide that. And then once this gets in, then you can do things like checking for duplicates. It will warn you if things are look like they've already existed. It will allow you to do things like see what the stereochemistry is, so you can see what the R's and S sites are, and so on. And there are a lot of little other tools that come along with this to help in the registration process. I won't go into all of them now, but down below you can add information that might link the substance to external databases, that might give relationships between substances, various notes, and even properties for the substance. So the forms help you enter in all this information from a user perspective. So after adding a chemical structure, you also can do some searches. You can analyze some data that exists. So in this case, I can copy the chemical structure that we just imported. I can modify it, and I can decide to remove certain sections of it to maybe help search for analogs. So in this case, I can say, let me find a similar steroid that has the same basic structure. I can do a substructure search and find all records in GSRS that have that motif. And the substructure gets highlighted. You can see all the various information we have about it. And you can even export all the records into various exchange formats. You can also do other kinds of structure searches, including similarity, exact flex. Similarity is based on the chemical feature similarity, just saying they have similar functional groups and layout. Exact and flex are more specific in looking at the either an explicit exact match on the chemical definition, or a flexible one that allows for some stereochemical differences and salt form differences. But there are also registration forms. There are registration forms for chemicals, but there are similar registration forms that go into a certain level of depth for each of the other substance classes. So if you're going to register a protein, there are there's information you would add about what the protein is. You would add what the chemicals, what the amino acid sequences are for each one of the subunits, and disulfide links to glycosylation and modifications. Similar things happen with other registration forms as well. And the software by itself allows users to have their own database locally to curate and maintain their own substance data. As you're using the registration forms, you can import a JSON file that someone may have given you from their own GSRS system. You can also show your own JSON and export that you could send to somebody else to have the definition, the full definition, together with all the information you see here in a importable format. So that's the basic overview of how the registration pieces work. But of course, once you've registered something, there's a lot you can do for browsing and searching. There's rich fielded text searches that you can do at very granular levels. Like we showed, you can do the structure-based searching. You can also do sequence alignment-based searching. So you can do things that are very much like a BLAST search for similar amino acids or nucleic acids. There's also one of the things we use the most at FDA, at least, is the customizable facet filters. These are those filters that show up on the left side when we're searching or browsing that allow us to quickly slice and dice the data to say, find me all the substances that FDA knows about that are proteins that have any kind of modification to them. It's there, and there's a huge number of these facets that we tend to use, and they're very customizable. So, we know that a lot of people using the software add their own, and it's quite easy to add your own. You can also have customizable data export formats. Out of the box, the software supports SD file exports, but it also supports the JSON exports, the Excel, and many others. But it's fairly easy to make your own exportable format as well, or with custom columns or whatever else you might want. There's a bunch of other tools that are worth mentioning for GSRS software. One thing to mention is just that there are administrative tools for user management, CV management, so control vocabulary management, and those have some interesting features that are worth discussing at some point, including things like registering sugars and linkages that might be used inside of nucleic acids. But there's also all the software, all the things you see in the UI, the user interface. Those are all based on a fully functional REST API. So it's a programmable interface that developers and integrators can use to connect the GSRS tools internally to their own systems. And we've made heavy use of that REST API internal at FDA to synchronize existing systems with GSRS without having to have direct access to the same databases or same source code. It's been very useful. There's also ability to make a lot of other customizations, custom scheduled jobs that run every night that make a certain kind of report or make a certain kind of data operation. There are custom triggers for doing standardization that are supported, and of course custom reports. But the other big thing to mention is that there are tools that use the REST API directly in order to do interesting bulk registration operations. So, for instance, on the NCAT site, they have a link to what they call the genus Excel tools, which are power tools that are useful for doing batch registrations to a GSRS instance using just a large spreadsheet with just a few fields, or getting out huge amounts of data from a GSRS instance just by having like a column of names and then selecting them and asking for the corresponding unique identifier. So the GSRS software, the open source software, ends up forming the backbone for a lot of existing software and substance related database projects. I've just highlighted a few of them here. There are, at least 3 systems. There are a few others that we know of, but that are in production that use GSRS as their base backing software. Usually, there's some customization that happen. The FDA's internal substance registration system uses this open source software with some customizations. Germany does something very similar with their own customizations, and then NCAT uses the software just as a read-only version for public consumption, annotated with some additional information about drugs. There are a few other projects that are approaching production, both the EU SRS and the United States Pharmacopoeia SRS, both internal to them, not public facing. Those are in process and are making good good progress. They seem like they will be emerging soon into a production environment. And then there are a few other projects that are in pilot phase. Larry mentioned that there is a collaboration with the WHO UMC that's mostly focusing on trying to capture and standardize vaccine ingredients, and they have an instance of GSRS that's being used to pilot this. And there's some exploration about where that can end up going, and what the limits of that might be. Similarly, there's another pilot phase project at FDA to use the Precision FDA platform to have a portal of GSRS that's publicly accessible behind a login that might even allow for some limited exchange of data in a more collaborative environment with regulators and with industry. This is just a high-level development timeline overview. The important thing to note here is just that the software has been in development for quite some time, and it's gone through a few different iterations. The current version that most people use is an offshoot of 2.7, GSRS 2.7, which was released in this spring. But there have been many other previous versions going in the past. FDA has used some version of the GSRS database since early 2017. Germany has been using it since 2019, and the WHO UMC group has been using a pilot of this since early this year. There are other projects also that are going forward along this way. The important thing to note is that this GSRS 3.0 is imminent by the end of this summer or early fall. So it's worth talking a little bit about what the software architecture of this whole these tools are. This is a very, very quick overview of it. Really, there's a core Java application that's built on the Play framework. That core Java application then makes use of some certain custom indexes for text and chemical information. It uses some custom plugins that can be customized, and then it uses a connection to some database agnostic database, and it can help you set up that database, whether it's in MySQL, Oracle, Postgres, or whatever else it might be in. Then it exposes the REST API, and either external applications talk to that REST API, exchanging some JSON messages, or the front end that you saw, the user interface, talks to that REST API. That front end is written then in Angular. The 3.x software architecture, 3.0 is imminent, as I said, is a fairly major rewrite where whereas the previous architecture had all the pieces of the software somewhat in one monolith with just little areas for customization. The 3.0 software architecture is much more of a microservice one, where there are specific entity services that are for substances and for vocabularies and for other things, and then there are isolated other microservices that do a certain task. And this will make the software a little bit more flexible for expanding and for maintaining. It also ends up using a more modernized framework of Spring Boot instead of the Play framework, which was what it had been using. The important thing to note here is that once 3.0 is available, it'll be very easy for someone to add their own custom entities that they can link to substances like clinical trials, products, and so on. And that way, those same high-level entities can be indexed and shared within the same GSRS framework, without having to have people have to have these extended entities, which the current framework would require somewhat. If you want to have other entities, everyone has to have those entities. So the resources that are available for this, the main place to go to is the GSRS NCAT's product site. This has information about the software, has previous presentations that have been given, and you can sign up to be notified. They even have a mechanism, I think, through email to get onto a collaborator Slack if you want to start slacking with the NCAT's team on GSRS. They also have information on the Swagger API, REST API documentation, and other things like that. The GitHub source code is here, and like I said, you can get on the notification list by going to the project site. I just want to give acknowledge this is this didn't happen by a single person or even a single team working together. Really, this is a very small section of the people who've been involved, and it's been a tremendous, a tremendously collaborative group who've worked really well together. And I only feel bad that I'm only showing these names because there are so many more that have deeply helped the effort, and it's really been the highlight certainly of my career has been interacting with all these people trying to solve these kinds of problems.
Thank you for those great presentations. We'll now transition into our Q&A session. If you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. We see some questions coming in right now, and the first question will be addressed to Ron Fitzmartin, and here's the question. Why does FDA not have a regulatory or legal mandate to implement IDMP standards?
Hi, thank you for that question. Why? Well, we don't have a regulatory mandate. Yes, that is true. Unlike the EMA that has a regulatory mandate and time frame for implementation, there's no statute, there's no regulation, there's no guidance at this point for IDMP. As many of you know, and as TJ probably went into a little bit, we have been conforming to these IDMP standards for many, many years. And so to date, we have not seen the need to have a regulatory mandate for conformance. We certainly support the IDMP standards, and we are working with our international colleagues for the time when we can have a global implementation. Thank you.
Thank you for responding to that question. The next group of questions will be directed to TJ Chen, and here is the first question. Will this change the NDC drug listing process? This is Ray from SBIA. It looks like we're having some connectivity issues with TJ's microphone. So what we're going to do, we're going to give an opportunity for TJ to check the connection. And we're going to move on to the next, our next presenter, Larry. Larry, and here is the next question for Larry. When we talk about pharmaceuticals, it has also included advanced therapy medicinal products, or ATMPs, such as Kymriah 1.2 times 106 minus 6 times 108 cells dispersion for infusion. Can you respond to this, please?
Yeah, hi, Ray. Yeah, we do describe those in the GSRS, and they are some of the more difficult things that we describe. We basically describe them, are they an autologous cell or not an autologous cell? If they're autologous cell, we capture that. Then we capture the transformations that make the cell, you know, if they're transforming with the vector, and then we also capture the expressed protein. So we do describe them at the substance level, and we provide an ID for them. But they, again, a lot of them are very challenging. We don't certainly know how many copies for some of the things that actually get into the cell. We don't know how many copies oftentimes get into the cell, but we try to capture as much information as we can at them and to distinguish them from other processes. Again, what we're trying to do with the Unicode is really distinguish things that we think are the same and, distinguish things that we think are different and assign different Unicodes to them. So even though they're different patients and they're different autologous cells, they would all get the same Unicode. Thanks, Ray.
Thanks for responding to that question. And get the next question for Larry also. Within substance identification, do we only look into API, or do we also classify excipients as used in the formulation?
Yeah, thanks, Ray. Yeah, we describe, as I mentioned before, we ascribe unis to all the ingredients that are listed in the product. That's required. When you list a product using SPL, all the listed ingredients have to have a Unicode. Excipients, of course, and there's been a number of questions on excipients. Maybe I'll try to address some. Excipients, of course, are some of the most complicated materials sometimes, and sometimes there are multiple substance ingredients. And, we've tried to, be consistent with the inactive product dictionary, and there's been a lot of cleanup and synergy between them in recent years, but it may not be complete. And, but again, excipients, if we give something the same uni, it doesn't necessarily mean they're pharmaceutical equivalents and can be substitutable for each other. For instance, in a polymer, that we may not capture all the aspects of polydispersity. So you can have drastically different functionality for things that have the same Unicode, but it's just that the elements that we thought were important for defining these things we've captured. And then for multiple substance ingredients, there can be a lot of ingredients that are there in small amounts but have a drastic effect. And again, we wouldn't necessarily, at SSG1, we wouldn't make that public. We don't make the public formulations, they still say private, the stuff that's in a DMF, and we wouldn't release that. But we would expect for Unicodes for all the ingredients that are in any excipients that you would have that and you would list that. Yeah, thanks again for the question.
Thank you for responding to that question. We do have another question for Larry, and here's the question. For the 10-character long substance identification, can you share with an example what characteristics we need to use to reach to this code?
Yeah, again, Ray, it's a random code, so it has no significance with the code. I mean, there's no information encoded in the code. We thought that was important because we didn't want to, if we give away the code, there was some thought that for things in clinical trials, we would have the code available, but not any other information. So there's no real information in the code itself. It's all random 9 characters, and then there's an algorithm, a fairly simple algorithm that gives the 10 check digit character. But, so the Unicode Unicode itself contains no inherent information, and that's was something we decided early on, that we wouldn't have a code that would have any. It's not a hash of anything. It's really just a random characters that are tied to the given material. And again, we try to tie the Unicode to, like Tyler said, to actual material. And even if we make a mistake in describing the material, miss something, that Unicode, when we add something to the material to further define it, it's going to still have the same code. So I hope that answers the question, Ray.
Thank you for responding to that question. We have one more question for Larry in this round, and here is the question. Can different manufacturers get different unis for their own versions of a substance?
Well, that's sort of a complicated question in some sense. I mean, certainly for a simple chemical, no, they can't. For what's meant by a version is always something that's difficult for us to know. And in Germany, for instance, like all the PEG polymers, they consider like PEG 2000 and PEG 4000 to be different versions of the same substance. We would consider them to be different substances. So again, for different manufacturers, it's not tied. The Unicode's not tied to a manufacturer. It's really tied to the material. And so different versions of the substance wouldn't get a different Unicode. And certainly if you change some critical parameter, critical characteristic, if you added a ligand onto a protein or something, that would then be a different substance and would get a different Unicode. But again, versions of the same material or batches, they all would have the same Unicode.
Thank you for responding to that question. The next group of questions will be directed to Tyler. And here's the first question. Does the downloadable GSRS software come with a blank database, or does it come with data?
Yeah, hi Ray. That is a great question. The software really comes blank by itself. If you download it, it's just its own blank software. But NCAT has made available a seed set of data that's based on open source. Public software, public data that's available to the community, and it's seeded with the uni data directly. So the answer to the question is, it does come with a blank database, but you can easily populate it with the public substance information that FDA has made available.
Thank you for responding to that question. The next question is for Tyler. How have small businesses used GSRS software? Is this software mandatory for working with the FDA?
That's another great question. I would start by saying it is not mandatory at all for use with the FDA. No one is required to use this software locally. What we would like to know more about how small businesses and industry in general are using the software. We do know that some companies decide to use the GSRS software internally to help with their own substance registration. We also know that some companies will just take it internally and then pull out certain pieces they find useful, like elements of the software, like the image to structure tool, or just the chemical rendering, and we'll sort of use those pieces together, and we allow that. We don't we allow them to do what they would like with it. So we'd love to hear more, though. We'd love to hear more from industry on how they're using it.
Thank you for responding to that question. And the next question for Tyler. How have small businesses used GSRS software? Is this software mandatory for working with FDA?
I think you just asked that one, Ray.
Let me go to the next question. I'm sorry, we had a duplicate here. Does GSRS software support other parts of IDMP outside of substances?
Yeah, that's another great question. At its core, the GSRS software is intended really to handle the ISO 11238 part of substances. Part of the IDMP. So it really is about substances. There are ways to interface this together with larger efforts to do IDMP, but GSRS itself is fundamentally still about the substances directly.
Thank you for responding to that question. We have one more question for Tyler, and here's the question. Does GSRS software guarantee uniqueness of substance definitions?
That's a great question, and the answer is no. The software provides tools for users who are registering substances to know when two substances are likely the same, if they are close match either chemically or through amino acid sequence or things like that. But they're really not. It really isn't a guarantee. There, as Larry mentioned, there's a lot of other things that go into defining a substance. And we really encourage folks to, when they're using the software, the same as we do at FDA, to have expert review involved as well to make sure that things are adequately defined beyond just using any automatic way of confirming uniqueness.
Thank you for responding to those questions. We're going to move back now to TJ Chen to see if we're able to establish microphone connectivity. And here's the first question. During your presentation, you mentioned that FDA has an IDMP implementation plan. Is this implementation plan publicly available?
Thank you, and that's a very good question. We are starting to implement or to draft the IDMP implementation plan. We do hope that when it gets more concrete, we will communicate and share some of the information. Thank you.
Thank you for responding to that question. And we have another question for TJ, and here it is. Will this change the NDC drug listing process in any way?
Thank you. That's another good question. Short answer is no. The requirement in the CFR 21 section 207.35 describe what changes require a new NDC. That requirement aligns with ISO NPID, that 11615 requirement for a new medicinal product ID. So no, the short answer is no. Thank you.
Thank you for responding to that question. We have another question for TJ. Is there an indication as to how FDA will roll out IDMP to industry, or will we see a transformation to the SPL process or something different?
Again, that's a good question. Like Ron mentioned earlier, there's no regulatory requirement for FDA to implement IDMP. Hence, there's no requirement that the industry would need to implement IDMP as per FDA direction. However, we have been using standards similar to IDMP for decades, and our goal is to continue to support that standard. And we do anticipate certain collaboration with EMA on the FHIR's message for the medicinal product information. We will transition, if we do transition to FHIR in the future. That is going to be a long transition. We're going to support both SPL standard and the FHIR standard for a period of time to allow a smooth transition. So the short answer to that again, no, we will not impose any change to the industry, at least not in the immediate future. Thank you.
Thank you for responding to that question. We have one more question for TJ in this round. For the messaging, will FDA also use FHIR as in the EMA?
Thank you. Yeah, that is, I think on my previous answer, I covered part of that. Short answer is we do anticipate to support or exchange medicinal product information with EMA in the future. And that would require us to support the FHIR, the FHIR message at some point. And we are collaborating with EMA during the HL7 FHIR development, and we are monitoring the whole progress. Thank you.
Thank you for responding to that question. We have one more question, and it's a little bit longer one. It'll be directed to Larry, and this could be our last question of the day. And here's the question. Concerning your concept for specified substances, many excipients which come from different suppliers and all meet the same specifications but are compositionally different in many ways also have a significant impact on the excipient's performance in the drug product. These components have concomitant components but are not typically defined in compendial or manufacturer specifications. Many times the presence of these components are considered to be highly confidential by the excipient manufacturers, and they will probably not provide these details except through a DMF process. How would your specified substance systems differentiate these different types of excipients?
Again, I touched on that a little bit before, Ray, but again, at the Specified Substance Group 1, we anticipate that most of that data will be kept confidential at FDA, that we won't release it. Whether the ingredient is listed or not, I mean, that's really up to the company. If they feel it's necessary to list it in a label, they certainly can. For certain dosage forms, it's required that you list all the known ingredients, like for injections. For tablets, it's not. The other thing is that I mentioned, there's another question related to this for excipients and other things that, there's nothing stopping your vendors from supplying the Unicode. The Unicode is something that's freely distributable, that the FDA doesn't copyright it. And again, your vendors should be able to supply you with the appropriate Unicode for a given set of material. And they can always come to us and ask for it. And but I know that's a problem with a lot of final product manufacturers, that it is difficult to determine which Unicodes or which substances to put in and what's the correct Unicode. But I think a lot of the manufacturers and vendors have lists of Unicodes to their material, and again, if they don't have it, they can come to us and we would supply that with them. Thanks, Ray.
Thank you for responding to that question. That was the last question that we have time for. And we want to thank all of our presenters for their very informative and timely talk, and also for responding to numerous questions that came in. A few closing reminders.